you ever wondered how our cells make exact copies of themselves? The secret lies in DNA replication, a crucial process that ensures every new cell has the same genetic information. DNA replication is the process by which a cell makes an identical copy of its DNA. This process is essential for cell division, allowing organisms to grow and repair damaged tissues. Before a cell divides, it needs to make copies of all its parts, including DNA. This way, each new cell will have everything it needs to function properly. Before we get into the steps of DNA replication, let's quickly review the structure of DNA. DNA has a double helix structure, resembling a twisted ladder. The ladder's rungs are made of pairs of nitrogenous bases, adenine pairing with thymine, and cytosine pairing with guanine, and the sides are composed of deoxyribose sugar and phosphate molecules. DNA replication begins at specific locations on the DNA molecule called origins of replication. In prokaryotes, there is typically one origin, while eukaryotes have multiple origins along the DNA molecule. Enzymes called helicases unwind the DNA double helix, creating two single strands. This forms a Y-shaped structure known as the replication fork. Before DNA polymerase can start synthesizing new DNA, an enzyme called RNA primase adds a short RNA primer to each DNA strand. This primer provides a starting point for DNA polymerase. DNA polymerase attaches to the RNA primer and begins adding new DNA nucleotides, synthesizing the new DNA strand in a 5' to 3' direction. Adenine is always paired with thymine, and cytosine is always paired with guanine. DNA polymerase has a proofreading ability, meaning it can correct errors during DNA synthesis. If an incorrect nucleotide is added, the enzyme can remove and replace it with the correct one. On the leading strand, DNA polymerase synthesizes continuously toward the replication fork. On the lagging strand, DNA polymerase synthesizes in short segments called Okazaki fragments, moves in the opposite direction of the replication fork. Once the entire DNA molecule has been replicated, the RNA primers are removed and replaced with DNA nucleotides by DNA polymerase. DNA ligase seals any gaps between the newly synthesized DNA fragments, create a continuous strand, completing the replication process. DNA replication is essential for growth and development, ensuring that new cells have the same genetic information as the parent cell, helping repair damaged tissues by producing new cell. Thanks for watching. If you enjoyed this video and found it helpful, please give it a thumbs up and subscribe to our channel for more educational content. And if you have any questions or topics you'd like us to cover, leave a comment below. See you next time.